EBSCOR, the experimental program to stimulate competitive research, is a National Science Foundation grant program. The goal of EBSCOR is to strengthen science and engineering research and education throughout the United States, and to enable states like Wyoming to be nationally competitive in these areas. EBSCOR aims to improve scientific infrastructure in Wyoming and build science, technology, engineering, and math research capacity across the state. One way Wyoming EBSCOR helps strengthen science research and education is through student programs such as the Summer Research Apprentice Program, also known as SRAP. SRAP is an intensive 6-week paid research experience for high school sophomores, juniors, and seniors aiming to attend college. It offers students a hands-on research opportunity in science labs across the University of Wyoming campus. Students work one-on-one with a faculty mentor throughout their time in the program. SRAP not only provides students with a paid internship and real research experience, but also helps prepare them for college life.
Okay, so next we have Ms. Makenna Fair from Boone, Colorado, and she'll be presenting How Does Density Facilitate Food Location? A comparison of visual and chemical cues in the New Zealand mud snail, Potamopyrgus antipodarum.
Hi, I'm Makenna Fair, and like I said, I did a study on the Potamopyrgus antipodarum. I worked with Dr. Amy, Chris, and Brenna Hansen in the Zoology and Physiology department. In this discussion, I will talk about the Allee effects, invasive species, our study organism, and our experiment. So the Allee effect is described as an increased benefit to an individual related to conspecific density. And conspecifics refers to same species. So this positive correlation leads to an increase in growth at lower densities. Shown in the graph here. So the Allee threshold is the smallest the population can be before experiencing a negative growth rate. So types of Allee effects are more mating opportunities because in higher densities you have more individuals, which means you have more opportunities to mate. Also, you have decreased predation risk. So for example, the starlings, a study on them found that they feed in higher densities because it means that they have more of a chance that at least one starling is watching out for predators the entire time they're feeding. And also they flock in the high densities because their predator, the falcon, it has a harder time seeking out a single prey. And then you have improved food location. So for example, hermit crabs, they forage individually, they spread out, but then once they find the food source, they all group around the food source. So then when they pass by the food source, it's visual, it's like visible to others. So then the timber rattlesnake, on the other hand, it uses chemical cues. So basically, it follows a trail of conspecifics, and even more impressively, It sits in the same spots conspecifics had sat, and even like they sit there, especially if the conspecific had fed. So these types of Allee effects can also benefit invasive species. And an invasive species is described as a non-native species that can cause ecologic and economic harm. There are 4 steps an invasive species has to go through to be considered invasive. So first, it has to have transportation from its native habitat to the invaded region. Second, it must establish in the new region by growing and surviving under the new environmental conditions. Then it must continue to populate and spread in new regions. So then fourth, it must alter the invaded region. So our invasive species is the Potamopergus antipodaria. It is the native mud snail from New Zealand, and it has become a worldwide invader. In the United States, it has invaded mostly in the Northwest and the Great Lakes region. So types, Potamopergus is considered a model organism for different types of research. It can tolerate a broad range of environments, which is part of the reason that it's been so successful in invading, because even though it's a freshwater mud snail, it's even able to survive in different salinities. And also, the Potamopergus in its native habitat can be found to reproduce sexually or asexually, but in the invaded regions, it's only found to be an asexual female. It's also known from other experiments that it benefits from high conspecific densities. Benefits of these high conspecific densities have been increased in reproduction, increased activity, and more likely to feed and spend more time feeding. So therefore, the purpose of our experiment was to find how the Potamopergus use high conspecific density to locate food, either via chemical cues or visual cues. Our hypotheses were feeding would increase at higher densities, and the feeding would increase in chemically conditioned water. We collected our Potamopergus from the lower Polecat Creek near Flag Ranch. That's up in the Grand Tetons. All of them were from the US 1 colonial lineage, and that's important because it means they're considered to be they're genetically identical. And then we housed them at the Red Buttes Research Facility and Aquaria, and they were given a mixed diet and kept on a 12-hour life cycle. For our experiment, we used the juvenile snails, 1.5 to 2.9 millimeters. They were provided a high-quality food and aluminum weigh boats. And for our experiment, we did 4 treatments and a full factorial design. So basically, our design, we did high density in conditioned water. For the high density treatments, we had 100 snails. And then we did high density in unconditioned water with 100 snails. The conditioned water came from feeding conspecifics. The unconditioned water was simply well water. The low density treatments was with 12 snails in the conditioned water and 12 snails in the unconditioned water. Each chamber consisted of 4 aluminum weigh boats that were placed next to the tubes that we used to inject the conditioned or unconditioned water. Every 10 minutes we would inject 0.2 milliliters of the conditioned or unconditioned water into the chamber. And then every 5 minutes, we'd count the number of snails in each weigh boat. So in our results, we did find that they fed in higher densities. And we also constantly noticed that they would feed when in the chemically conditioned water. However, the number of snails that would actually feed, really wasn't that high a number considering how many snails we have. So for our results, we did the mean number of feeders for the high density and low density, and it was significantly different. So when we compare them proportionally, you might think, well, of course, there's going to be more feeding because there was more of them. So we did compare them proportionally, and it was 10% would feed in the high density, but only 5% fed in the low density. So comparing high density unconditioned water to high density conditioned water, it was continuously higher, but not significantly higher. So therefore, we don't have the statistical power to show. And same for low density unconditioned water with low density unconditioned water. It was slightly higher, but we lack statistical power. So in our experiment, we found that Phytomopergus are more likely to feed in high conspecific densities, which was consistent with other studies. We found that this may be facilitated by chemical signals from feeders, but again, very few animals fed in our experiments, and we lack statistical power to show this. So future directions from our experiment are we need more experiments with the juveniles to demonstrate that the Phytomopergus use chemical cues. Also, we need to repeat this with adults because in invaded regions, it's not just juveniles, it's also adults. And also, it'd be interesting to do this with different types of invaders to see how chemical cues, high density, and Allee effects affect their feeding. So I'd like to acknowledge the Wyoming National Science Foundation EPSCoR for funding my research. I'd like to thank Lisa Beta and the staff, Dr. Amy Chris and Brenna Hansen, Joe Bobbitt and Steve DeVries at the Red Buttes Research Facility, Matt Caroline, Jacob Heen, Jonathan Rader, and Brady Goodman. Any questions? Yeah.
What ecological environmental effects does the invasive nest have wherever it goes?
It's found that it eats a lot of the native algae, and that it also releases ammonium, so it has an effect on the nitrogen cycle. Any more questions? Yeah.
So I see in the distribution map of the invasion, like you said, it's up in the Great Lakes, and then you see another chunk of it out in the west. What keeps it from going to the other parts? Do they know that? Temperature, water?
Well, right now, there's a lot of work being done by, I think it's the Division of Wildlife, because it's been found that these guys are transported either through fishing gear or the ballast of boats. So there's been a huge effort to making sure these guys aren't transported. So that's part of the reason they haven't continued to spread.
Yeah. Did you put more snails in the high density and less in the low?
We used the numbers because they compared to, like, the 100 snails was equivalent to approximately 2,000 snails per square meter, and the 12 snails was equivalent to approximately 250 snails per square meter.